So the booster trial was originally designed to be able to harvest archives from patients with lung cancer, early lung cancer, so that complete profiling could be done before and after surgery in order to discover biomarkers of the disease and to see if there were diagnostic as well as prognostic biomarkers that could be used for the monitoring of the disease. So my role in helping to design that really depended upon my ability to know how to do archiving. And I've been doing archiving for the last 30 years since the National Cancer Institute and have established a nice archive that I think reflects clinical stage 1 lung cancer, and that's what I presented at the meeting. Right. Amir became the principal investigator for Booster, and he will be running the trial, and we will be a participating site in the United States so that we'll be able to collect specimens for the trial. I think that those specimens will be collected under a given standard operating procedure that we've been using for the last 10 years at NYU, which ensures that all of our specimens will be collected and archived and processed uniformly. Well, it was 4,000, and the question is whether that's too ambitious and whether it's going to be 400. It's really going to depend upon how many sites will commit, how many sites will actually do their accrual and do what they promise they're going to do. And then also it's going to depend upon the quality of the archiving. And so the archive is one part, but having the clinical demographics to be able to go along with the archive is just as important. Well, I think that the present technologies include cell-free DNA, proteomics, looking for certain protein profiles that can say that a lung cancer is present, and then you can look postoperatively to see whether that profile is gone or changes back to a more normal profile. And then you can also look at the cellular content of blood, including the peripheral mononuclear cells, whether they're isolated by a Ficol gradient or whether it's Buffy coat. So there's a wealth of opportunities, not only genomically and proteomically, but also epigenetically, to look at different platforms to look at this archive. Correct. RNA is of great interest to myself as well as microRNAs, but I think that RNA from the blood, if you use certain tubes called Paxgene tubes, it'll be preserved and it'll be well preserved, but it's not as good, in my opinion, as blood that's cryopreserved or snap frozen at the time and not have to have it in a preservative. I think it's very important that the institutions that are part of the WINS consortium commit to that. I think they will need to really have a good working relationship between their pathologists, their surgeons, as well as to have a system already in process so that the specimens will truly be of value, because the quality control of the specimens will be of utmost importance for something like this. So you can take the curve, the survival curve of the worst of the stage 1 lung cancers, and you can look at time to progression, one of the subcohorts. Not every one of those patients progresses. Therefore, not every one of those patients dies of lung cancer, but yet they have a picture under the microscope that looks like a very bad tumor. So obviously there must be differences between that subgroup of patients who are going to do well and patients who are not. Well, you don't want to give a potentially toxic therapy to patients who you know are going to do well. despite the fact they have this other characteristic. So I think it's very important that we be able to, as mentioned at this conference, sub-categorize different groups molecularly, either by RNA or other ways, to be able to determine which ones need further therapy and which ones do not.